Good afternoon, everybody. I'd like to, it's actually my great pleasure to welcome you to one of the most exciting events on the Purdue entomology calendar in any given year, and that is the John V. Osmun Alumni Professional Achievement Award in Entomology. I would like to welcome some very special guests. We've got Dave Miller here. Dave, and Jay. An alumnus. We've got Tom Turpin, Professor Emeritus. And I'd also like to welcome, it's Jordan Lee, correct? Yes, welcome Jordan. Some other guests to welcome, some budding entomologists, Michael and Mia. Li-Byarlay, our junior entomologist is Hongmei's children, and her husband Wayne. Welcome. Our very special guest, our awardee, of course, Dr. Hongmei Li-Byarlay, and I'm going to say a few words in a moment. And also joining us from Central State University, and this is a real pleasure, Wayne, Kiana, and Cara, and, Joel, Cara, and Kiana, is that correct? And Danielle from Central State University. I know you came over this morning, I'm sure it was a long drive, and we had a great poster session with you this morning and really enjoyed that, so thank you, and I hope you've enjoyed your visit to the department. Okay, so I think many of us who had the pleasure to meet John Osmun really know the legacy that he provided this department and his commitment not only to the study of entomology and to advancing entomology, but also his commitment to our alumni. And you know, that is really the spirit of this award, is recognising the outstanding achievements of Purdue entomology alumni. There's been a long list of award recipients since 1988, and they're listed here in the brochure, in the program that you would have received. And this year's awardee, it is our great pleasure to introduce Dr. Hongmei Li-Byarlay. Actually, not there in the corner. I think many of us know Hongmei very well. I remember when Hongmei started at Purdue not that long ago. And she was a student in Dr. Barry Pittendery's lab, and I remember her training here at Purdue, and boy, she has come so far since that time. So her Bachelor's of Science and her Master's of Science degrees are from universities in China, her PhD from this department in 2007. And then she subsequently went on to undertake postdoctoral fellowships at the University of Illinois and the University of North Carolina. And then in 2007, she joined Central State University as an assistant professor and was promoted last year to associate. She's leading some groundbreaking research on the study of honeybees, honeybee genetics and genomics, and really blends beautifully both the basic and more applied aspects of research. She's the recipient of multiple funding awards from USDA and also from the National Science Foundation. But the other thing that really stands out in Hongmei's case is her exceptional commitment to the training of junior scientists, some of whom she's brought with her today. But if you had the chance to review her CV, you'll know that she has trained many scientists through her program and in research, and I think opened those scientists up to the opportunities that exist in entomology and research more broadly, and particularly to training underrepresented students. So. Which is a phenomenal contribution, Hongmei, providing those opportunities to people. So I know that we are going to be looking through the molecular lens with Hongmei's talk. So Hongmei will give a presentation highlighting her research and other, I think, academic and professional achievements. and then we will confer the award. But welcome, Hongmei, and congratulations. It's lovely to have you back again. We saw you a little earlier in the year, and can't wait for your talk.
Sure.
Thank you.
Yeah.
Excuse me, I'm just going to...
Thank you so much, Kate. It definitely feels so good to come back since I come back home. So since I have 45 minutes, and now I'll just start hopefully. Well, before I start, I would like to say thank you to the department, Purdue Department of Entomology. I'd like to thank my advisor, Barry Piedron, for the nomination, and also the committee for this award. And when I received Kate's email for the award, I was so thrilled and jumped off the chair.
She practically jumped through the email, I think.
So, well, without further ado, I'll just start my talk. I would like to introduce my talk by starting Charles Darwin's quote, such as simple instincts as bees making a beehive could be sufficient to overthrow my whole theory. So honeybees are one of the social insects a lot of scientists study, and it's a really fabulous model system for animal behavior. So Tim Morgan's animal behavior as a pioneer since 1963 had two essential questions. Either we ask a question in proximate questions of understanding the mechanisms, causation, and development, ontology of behavior, or we can ask the ultimate questions of the understanding of function and evolution of behavior. So today, throughout my talk, hopefully, I wanted to give you some case studies, tell you 3 different stories, and tell you about the knowledge we try to break through and try to gain more knowledge on what's underlying, what are the molecular mechanisms underlying the behavior. So these are the outline of 3 stories I would like to tell. First one is one recent published paper in behavior epigenetics. The second one is on pesticides and applicability. This is the paper we're working on, trying to submit pretty soon. And the last one is a current research ongoing on the landscape ecology and the foraging of the honeybees and other pollinators. So to start the first story, we're looking at aggression behavior in the honeybees. So aggressive honeybees, such as Apis mellifera sculata, escaped from Brazil back in the 1960s, are extremely aggressive. So here's an aggressive honeybee look like, and here's a European honeybee, Apis mellifera, most time linguistica subspecies look like. So when you kick a high box of EHB, maybe, a few bees come out and try to sting you. But if you stand by AHB hive and you kick a box, and hundreds of them are gonna get on your body and sting you. So that we're looking at the really different scales of aggression and defensive behavior from these 2 different subspecies. So there's one way to study aggression is to use reciprocal process. This is also a method that Professor Greg Kant here at Purdue University also used in the past many years. So the approach is here is to use either aggressive honeybee as the dad, and the European honeybees as queen as the mom, and then do artificial insemination. You have a F1 generation with a European mom and aggressive paternal, so M for maternal, the P for paternal source. So the F1 generation will be EA type. The other cross, you can use the aggressive honeybee queen as the mom and maternal side, and then the European honeybees as the paternal and the dad. And through the experimental insemination, we'll get aggressive maternal and the European paternal. So the interesting part is that through the research from Dr. Greg Han and Osmun work from the 1990s and 2005, there's a really interesting phenotype. One, there's the European maternal and aggressive paternal F1 generation, they see a higher aggression behavior. So that indicated the aggression perhaps is influenced by the paternal source in this way. So there's a paternal effects on the aggressive behavior. So when we look at the genetics of honeybees, it's a haploid-diploid system. The females, for example, there's a queen here, they have 2 sets of chromosomes, and all the workers as well have 2 sets of chromosomes. And each set is 16 chromosomes. The male is only 1 set of chromosomes. So with the haploid-diploid system, which means it's quite interesting, all the drones will only have which means I'm trying to remember. So the drones have daughters, but they will not have grandchildren. Is that right? I'm trying to remember. So anyway, so because the haploid-diploid system, we have the potentially looking into the parent-specific gene expressions. So we can sequence the honeybee genome and look at what are the genes from paternal source and what are the genes from maternal inheritance. And by comparing these genes, we can study how the father and the mother influence the offspring. In this particular case, we wanted to study how they influence the aggression behavior. So in sociality, there's different theories. There's a kinship theory and there's a group theory. In the kinship theory, I pointed out the altruistic behavior supports relatives. So what that means is within the hive, the relatives will help their relatives, super sisters. And there's also a conflict between the paternal and maternal effects in a group, right? So there's almost like a computation from the paternal side and the maternal side. And both father and mother wants to invest in the offspring, and they wanted to maximize their investment for the next generation. So if we can study the parent-specific gene expression as well as their parent-specific epigenetic modifications, these are chemicals, modifications in the genes, I'll talk a little bit more. And then we can study the pathogens that are from the dad, how these pathogens may influence the aggression behavior. So there are a lot of different epigenetic marks on DNA and also in RNA. And our goal for this project is to look at the M6A. So this is the N6 methyl adenosine. This is called the M6A RNA methylation. So this type of chemical modification is on the RNA. And what happens is that it can add methylation. It can add a methylation mark into the adenosine and make it methylated. And this is most abundant in eukaryotic RNA modifications. accounting for almost 80% of all the RNA methylation. And in humans, in honeybees, in a lot of insects, and they all have RNA methylation. However, there's really not much known about RNA methylation in the honeybees. Last year, in 2021, the first paper published has revealed that honeybees have RNA methylation. And there's also, they have studied the queen and workers, and they look at different genes such as vitellogenin, the insulin, lipid protein, insulin-like protein 2, and the juvenile hormone peptides are associated with the difference in the RNA methylation. So, because the important roles of M6A RNA methylation in different insect species, besides honeybees, we also know in silkworms, they're having studies to show they're involved in transcription and translation. In the Drosophila fruit flies, M6A is also associated with behavior, such as walking behavior and learning memory. And with all these evidence, I think it will be really interesting to study how M6A methylation can be involved in the behavior, social behavior in the honeybees. So these are the mammalian model research. We know that M6A is associated with the cell differentiation, inflammation, and a lot of cancer studies. So genomic imprinting is another interesting topic. The crosses between the same genetic strains can help to study genomic imprinting. In humans, we know the genomic imprinting have the maternal imprinting genes from, everybody has some genes influenced from your mom and then your grandparents. So there's a parent of origin effects in the honeybees, we know, but we don't know anything about RNA methylation. And so, so we think with this research, we can also try to provide more information to go into look genomic imprinting, whether honeybees have imprinting genes and what they look like. So this is a National Science Foundation funded research, and so this is my graduate student, Alan Lee, and this is another graduate student from Penn State, Christina Grosinger's lab, and Sean Bransnahan. So both of them worked on this project. We started back in 2020, and with the 2 COVID year we paused, but we still, with the 2 years of field data, we're asking this question whether there's any change in the parent-specific gene expression and whether the M6A RNA methylation has anything to do with aggression. So we first started with the behavior data, and this is the data from 2019 actually. So this is also a collaboration with Penn State as well as Juliana Rango's lab at Texas A&M. So in Texas, we're able to do these crosses and instrumental insemination. When we have reciprocal crosses, we get the EA type, as I mentioned earlier. So those are European paternal, maternal, and aggressive paternal. Or you can have the aggressive maternal and European paternal. So with all the trials of aggressive behavior, we set up a colony and then we banging on the table of the colony. We also have a leather, piece of leather with alarm pheromone in front of the hive, and that will really stimulate, irritate the hive, and the bees will come out. Then we measure how many stingers are around the leather. So both of them together, all the 4 trials together, we see a significant higher aggression, which is consistent with Dr. Greg Hunt and the work published before. And then, so in 2021, we are at Penn State. So these trials are at Penn State. We tested in different, besides the European honeybees and the aggressive honeybees. We also testing the Cordovan weaver and the weaver Cordovan type. So again, maternal and paternal on the top corner. And we see a similar trend when the weaver bees are actually from Texas. And even though they don't claim they're aggressive, I think potentially all the bees in Texas are aggressive with the trend. So we see again the so actually, yeah. So with the weaver as paternal, we see yellow, and then the green one. So actually, I think in this year's data, we didn't see the trend we would like to see. And then the other process we have is the Cordovan maternal with a Penn State paternal, and then the Penn State maternal with a Cordovan paternal. So with this one, we see also. Oh, okay, so I remember. So these colonies, we're testing them without the aggression behavior context. So purely, we just want to see the paternal and maternal effects in this way. So I think that's why we see the similar trend here. But these data are not published data, and we're still trying to work on these data sets and see how we can design the further experiment. So with the 2019 data set, we are able to do sequencing. So we sequenced the aggressive bees versus control workers from the hive. So these are the inseminated colonies. They're either the maternal European, paternal aggressive, or the other process. So we collected workers that are either aggressive when we banging on the colony, when we did the behavior assay, or the control workers that didn't respond. And from both processes, we collected at least 6 workers from each group. And then we did a transcriptomic analysis, RNAi, and then also a methylome analysis on the RNA methylation study. So when we look at the parent-specific gene expression, we can see the different colors here. So the blue color coded for paternal bias genes. The yellow color are the European honeybee bias. The green ones are the aggressive honeybees bias, and the black ones are the maternal bias. So without this, we can see a significant difference here from the differentially expressed genes. So all these genes in the color means they're parent-specific. So we did a geo-analysis, look at the mitochondria, the genes involved in mitochondria, energy, or the cellular skeleton organization. And so here is the data of the RNA methylation. When we compare the, again, paternal bias, the maternal bias, and also European honeybees and the aggressive honeybees, we see less number of genes here. So that means indicated there are less differentially methylated genes detected. And then we did a statistical analysis, and we didn't see the significant difference here. So differently spliced genes is another way to look at gene regulation. And because the genes have different exons and introns, when they have maturation of the messenger RNA, they can keep certain exons and remove the introns. But within the cells, there are different signals and cellular activities. There can be different ways to splice these genes, and then you get different type of transcripts of the mRNA, messenger RNA. So So when we look at the differently spliced genes, we see the genes involved in synaptic physical, adenine synthetase, exchange transporter protein, hypoxia protein related. So I think in discussion for this part of work, maybe I'll just fast forward, go fast on this. So I think we detect the dominance for aggression behavior. We definitely detect significant difference in the gene expression levels from either paternal or maternal origin of specific genes. The paternal effect as part of the intragenomic conflict may invest in the social behavior. So the fathers and the paternal investment may maximize their input into the F1 generation in the offspring. And meanwhile, they may allow for some more specialization in the defense behavior. So there are also parent-specific gene expression identified from the nanopore direct long reads. So I'll just pass this part. So I want to come to the second story. This is a new research. We're collaborating with my collaborators in China. It's the Hunan University with with a PhD student Ma Changsheng. So in this study, we use fly mills to study the flyability. So honeybees can fly with their wings, and they can fly actually quite far. If you have a colony, when there's needs for foraging to go to collect nectar or pollen, they can fly up to even 5,000 meters far away. And so for these bees, the flyability is important because if they can fly well, then they can forage well for the colony. They can collect food for the colony to survive. So in this study, we are interested in different types of pesticides with a field level dose. We wanted to know how these pesticides can, whether they will affect the honeybee flyability and how will they affect the honeybee flyability. And these are tested in the Apis cerana species, so that's Asian honeybees. In the video here is a fly mill. We actually have 10 fly mills in our lab at Central State. So this is an example of how the bees fly out there. So these are the 3 pesticides we tested. One is neonicotinoid, imidacloprid, and it's 10 nanogram per gram. The second one is organic phosphate pesticide, chloropyrifos. The last one is herbicide, that's glyphosate. Glyphosate is 60 nanogram per gram. All of them are mixed with a 50% sucrose sugar water and containing either one of them, 2 of them, or all 3 of them. So the fly mill can give several different data we can look at, including the maximum velocity of the flight, the mean velocity of the bees, the flying duration of the bees, how long they can fly, or the flying distance, how far these bees can fly. So all these bees tested, each treatment has 62 to 63 worker bees, and they're all managed for the same age. And the first 2, the max velocity and the mean velocity, there's no difference in these among all the treatments. However, when we have the flying duration and the flying distance, we can see really significant drop on the IC treatment. So that's imidacloprid, and the chlorophyll here. So, and also for the flying distance, we also detect the IC treatment. Both two of the pesticides together mixed cause the lowest flying distance. If we zoom in the last table, we can draw out all the lines for their flying distance. This is how far these bees can fly. And you can see the lowest one is blue, and blue is the IC treatment. So that's again the neonicotinoid and the organic phosphate pesticides together. So we also did an RNA-seq analysis. We look at the gene expression of the IC treated bees and how what genes are differentially expressed. On the list here, so this is a volcano plot. You can see all the red and green ones are significantly upregulated or downregulated. And among all those genes, when we did the gene ontology analysis, we see on the top the biological process. They're involved in G protein coupled receptor, signaling pathways, a lot of lipid metabolism, lipid transport. So lipid is important in the fatty acid of the honeybees, and it causes the energy. It basically is the source of the energy. For the molecular functions, again, it's involved in the G protein coupled receptor, the active transmembrane transporters, and the lipid transport as well. So we also look at the KEGG pathways. When you have all the gene ontology genes together, you can look at what are the cellular pathways that that are being affected the most. And the biggest circle here is metabolic pathway. So that means this metabolism in the honeybees are affected. Also, the next one is about synthesis of amino acids. On the red ones are increased, on the blue ones are decreased. So there's additional metabolic metabolomic analysis from the HPLC, and 17 of the metabolites were detected from the comparison of the IC treated versus control groups. And then we can see this is oleoglycine. It's definitely the most interesting because it's an acyl amino acid that is important for energy production in the bees, and is directly involved in mitochondria and their functions for energy. So summary of the fly study, we detected the imidacloprid. The imidacloprid and chlorpyrifos are together have the most severe effect, toxicity in the flight distance and duration. We detected 300 some differentially expressed genes. A lot of them are involved in the metabolism, lipid metabolism. For metabolites, there's 17 of them. So definitely these are field relevant dose, and we think they're very important to know. And overall, the explanation is that these two synergistic pesticide effects can influence the metabolic pathways in the energy regulation. So the last one is a current project we're ongoing, and this is a USDA supported. We want to look at different landscapes and farms and how the honeybees and small carpenter bees can have a different stress from these different landscapes. So abiotic stress, in this particular project, we're looking at pesticide effect here. The abiotic stress, we're looking at the viral stress here. And first, during the lunch, and graduate students were asking me, why do we include small carpenter bees? So the original proposal, we wrote it for honeybees, and then the reviewers and panel suggested adding wild pollinators. That's why we know more about small carpenter bees than any other wild bees, so that's why also collaborators with Sandra Rehan, Dr. Sandra Rehan at York University, and also Ree Johnson at Ohio State University. So our goal is to look at the mite resistance, larval stress, and biotic stress of viruses, the pesticide residue stress from the pollen, as well as the habitat colony management, how we can help the organic beekeeping or help the improve the beekeeping practice. So there are 3 farm landscapes we try to focus here. The organic farms. This is one of our farms in Cadiz, Ohio. This is conventional farm. It's in Clayton, Ohio. This is an example of roadside. We want to use roadside as a control. So they should not have a lot of pesticide residues in these control sites. At minimum, we're looking at just herbicides in there. The experiment design is to place honeybee colonies as well as small carpenter bee nests in these farms. And then we collect their adult bees, larvae, pollen, and we look at the pesticide residue from different farms. So these are examples. This is Joe sitting in the chair last year, in the summer. So we have students work on several students help on this project. We used old raspberry cans to make them as stem, tied with the bamboo sticks together, put in the field, and then we are hoping the small carpenter bees here, like Serotina, is the example, will go into the nest at the beginning of the field season, and then we collect these nests. So my students have opened many of these stems. This is an example. That's pollen balls of small carpenter bees, and these are young larvae or egg just on those pollen balls. So in June, we're really working hard to try to collect all these pollen balls and before the larvae eat them. So from the pesticide study so far, we compared honeybees versus Serotina, and we can see that Serotina, small carpenter bees, have much less pesticides in their pollen. And for example, their pesticides, boxacalid, their herbicides, atrazine, metolachlor. These are also reported from other studies in blueberry farms as well. When we look at the honeybee side, you can see a lot more pesticides, herbicides are in there. Some are fungicides as well. When we compare different farms, we compare conventional farms versus organic. So ideally, we should not see anything in the organic farms. However, in reality, some of our organic farms are not gigantic big. So I tried my best to try to find all the organic farms you can find to work with us near our campus. So from these results, at least still, I think when we look at the list, there are more pesticides, herbicides come out of the conventional farms than versus the organic farms. So the reason for these, some of them showing organic farms, is because outside of our organic sites, there are other conventional farms in there. So my student Kiana also helped. She started last November to help on the project and to look at oxidation stress. So oxidation stress is a marker to measure how healthy the cells are. And so it's highly related to aging. when we measure the oxidative stress, we can have some idea the status of the bee health in the colonies or in the small carpenter bees. So we have compared organic bees. These are adult honeybees from organic farm, roadside, and conventional farms. And we did an MDA assay versus total protein. So MDA is a marker for lipid peroxidation. And we're measuring the lipid damage by measuring the MDA. And kind of surprisingly, the result is not what we expected. I think our hypothesis is that conventional may be the highest, right, with the highest oxidative stress. However, we see the roadside actually display the highest by statistical analysis, so this is more significant than the other two sides. And then we look at the larva, we see the similar trend. Again, the roadside has experienced the highest level of oxidative stress here than the other sides. So we also, Kiana also worked on the small carpenter bees. Small carpenter bees are really small. At the beginning, we were not sure how they will, turn out in the assay, and we try to group them or individual ones, and eventually we think the individual ones work. And the results are kind of similar to the honeybee ones. Again, we see the highest in the roadside, and it's significantly different from the organic farms, but it's not significantly different from the conventional. So in summary, we still have more data coming. We are hoping to get more data from the pollen identification as well. But from so far, the data from pesticides and from the oxidative stress, we can see that honeybees definitely, the honeybee pollen has a lot more pesticides, chemicals, residues in them. One reason to explain this is perhaps maybe because the foraging distance and the range is way bigger in the honeybees. They can fly further, far away to collect pollen and bring back to the hive. As I mentioned, maybe up to 5,000 meters. However, the small carpenter bees are small. They're much smaller than the honeybees, so their flying distance and the range will be smaller as well. So that may be one reason that's why less flower diversity in the small carpenter bees, and that may explain why there's less pesticide residue detected. The pollen from conventional farms has more pesticide residues than the organic farms as expected, but we didn't expect organic farms can have several pesticide residues. And the last is the oxidative stress level. The roadside has the higher level of oxidative stress than the organic farms or conventional. So one reason to explain this is that besides the pesticide as the environment factor to the oxidative stress, there are also other ecological factors, and there is a community in the roadside environment. So besides pesticides, there are also other insects, other plants, other ecological factors that may also cause the increase in the oxidative stress. So overall, I hope I showed you the 3 different stories of our research. And within the last 6 years working at Central State University, I wanted to really help students to be successful. And we are focusing on scientific training of all the students, leadership training. Every summer, we run a 6-week-long workshop with a coach and teach students about leadership skills. We encourage them to write a successful story to themselves and to the group, and to think about try to encourage them think as heroes, to go to do summer research and to do research with our lab, and we want them to feel good and feel that they have achieved a lot. So with the mentoring and teach them about life, I hope we cultivated a lot of really bright minds for the next generation of students. So this is just a short video to show you this summer. We have, so this is our end of summer presentation. So all the students, we have 5 students presented their research. Also our postdoc presented. Everybody received a certificate for their leadership training. That's our picnic at the Cedarville Park with a waterfall. So, yeah, with that, yeah, this is a new project we just started. We're hoping to initiate a new graduate program with a master, to start with a master program in both soil science and entomology, and to train more students in agriculture. And we also want to take students to do more internships with, for example, USDA agencies, the industries, and the government, to train them, know more about agriculture. And last, we're doing K-12 summer camps. We're planning to do middle school, high school students engagement projects in the next 5 years. So I'd like to thank all the funding, my family. So I'd like to thank my husband, Wayne, my kids. They gave me unconditional love and support me, support my career. And there are, of course, up and downs along the way. And now just out of play. This is my last slide. So I want to say I love Purdue entomology. I think at the beginning of my talk I got too nervous, so I kind of bumped into some parts. But I was able to find a picture with John together, and back in 2000, I'm trying to remember, 2006 maybe, 2005. So I really appreciate this award, the alumni award that John and his family started. I'd like to thank all my teachers. So I took classes from all of them in the past, and then my ITA for all of the great professors here. And so if I learned how to do entomology from all of them, I definitely learned how to teach from all of them. And they showed me a whole new world of how to teach entomology with fun activities, with the funny jokes and hilarious things. It's basically halfway to a comedian, right? And of course, I want to thank my committee members, Dr. Barry Pietering, Larry Murdoch, Bill Moore, also Dick Shea. I couldn't find Dick Shea's picture online, so we just have all of them here. And last is all my cohort, all the graduate students, when we were there together. That's all I want to say. So, love entomology. Hope you spread the love to more people. Thank you so much.
There are some characters on that slide.
So we can turn into an open mic.
So in the true spirit of the Osmun Award, we make you give a talk before we award you confer your award. So I'm actually going to ask everybody to hold their questions until we confer Hongmei's award to you. Have a good one, I've also realised we're missing John Obermeyer today.
Yeah, he's a broad, I believe.
He's abroad?
Yeah.
Well, Hongmei, I know that we have been hard at work to get this very special plaque. And maybe actually, would you like to come to this? Oh, wait, we're at the top.
No, I'm following you.
So it gives me great pleasure. 2023 John B. Osmun my Professional Achievement Award. Hongmei, congratulations, you're highly deserving, and we are so proud of you.
Thank you.
All right, now you can ask your questions.
Okay, so before you
I'll hold
ask
this for you. I'll hold this for you. Okay.
Well, yeah, before you ask question, I would like to give a department a gift. So today is actually our Chinese holiday. It's our Chinese Mid-Autumn Festival. The moon is as warm as it can be right now. So this is special moon cake. Means get together as a family and reunion. So coming back here is coming back home, and I wanted to give you this gift and the department. Yeah.
I'll exchange. Thank you, Hongmei. You'll always be part of the Purdue Entomology family, and it's wonderful to welcome your family back here today. I'm sure your kids are so proud of you, as well your students and folks from your lab. This is absolutely perfect. I think it was a full moon last night. Right? It was gorgeous, and you really, epitomise the spirit of the John B. Osmun Award. So thank you, Hongmei. I know we are watching you. You're a rising star, and I know you're going to do amazing things. Continue to do amazing things. So congratulations.
Thank you. Yeah. I'm ready for questions.
Now you can give her questions. Do I have to share this with I
There's more pieces, so it's up to you. You can cut them into smaller ones or just eat all your
Your slide is great because they're just so, it's a bit of a walk down memory lane with some of the folks there and some that are no longer with us actually. Yes.
Yeah.
And great that you have a picture.
And there are a lot more I think I also interact with and work with before, but I thought I'll just list all my teachers and my mentors here first. So sorry if I missed anyone else on the slide.
Questions for Hongmei about her research? I have one. Here we go.
I have a question on the landscape and pesticide interaction. When you placed honeybee colonies in the farms, were they started with new comb or old comb? Because
They start new comb.
new comb.
Yes, so all the pollen and also bee bread were collected in new combs. And we mark those combs, and so we know they're new. So every year, yeah. And just to avoid contamination of other pesticides from the old combs. That's why.
And I had a question on the alternative splicing.
Yeah.
If I understood correctly, you were looking at alternative splicing between bees in the same colonies or family units, and I was wondering how does alternative splicing work if they all share the same chromosomes?
So we're looking at the parent of origin ones. So we know they're from sex specific parent, but at the same time we want to look at all of their splicing patterns. So yeah, even though they're in the same colony, when in the cell within the cell, there's transcription happening, and when the transcription happens, there are different signals in the cell. And so there are certain, you can think of 1, 2, 3 different exons. And so you can have 1 and 3 exons together as one transcript, or you can have 2 and 3 together as another transcript. This is why there are different isoforms of transcripts. And this is why when it comes to translation, you'll see different isoforms in protein. So even though they're from the same colony, but during the splicing events, they can still happen. Yeah.
Do you have hypotheses as to what might cause alternative splicing if the bees are identical genetically?
Yeah, so one hypothesis behind it is the epigenetic control. What we were hoping is to find the differentially methylated genes that are also involved in the differentially spliced genes, like overlapping on the gene list, but I don't think we find any overlapping one. So we know in DNA methylation, it's been studied that the different DNA methylation marks on the different exon can cause the pause of the exon to be transcribed, and that it's one way to give a signal to the cell. So it's almost like try to make a simple way to tell you like a green light, stop, a red light, right? So if this exon has, say, less methylation, then it's a green light. Go ahead and you can transcribe it. But if this exon has too many methylation, then it's like a red light. Like, oh, don't transcribe this, jump to the next one. So we know in DNA methylation, these type of mechanisms happens. But for RNA methylation, so far we don't know anything about it. And that's another reason we want to study the splicing and the gene expression and the methylation. So ultimate goal is to find the genes that are in all these categories can explain why these paternal effects on the aggression. Yeah. But another interesting thing is that from the nanopore, these are nanopore long reads, we detected a way less alternate splicing isoforms when we compare to the RNA-seq Illumina reads. So I think one reason is because the long reads of nanopore give perhaps more accuracy on the sequencing, and there's less false positive when we align these genes into the reference genome. So there's maybe less false positive on alternate splicing. Yeah.
Thanks.
Yeah.
Other questions for Hongmei?
Can you explain the results of the roadside numbers? Why you think those were higher than the others?
So, as I mentioned earlier, on the roadside, we expect to be just minimum herbicide. So it's almost like a natural habitat for the bees. However, we detect the highest oxidative stress. We have another study I didn't present here. It's the study of feral honeybees versus managed honeybees. And we detect a similar trend. We thought the feral colonies, the feral bees, within the survival experiment, they actually live longer. So we thought that maybe they have a lower oxidative stress. However, they have higher oxidative stress compared to managed bees. So there's one way to explain it is that in the feral colonies, there may be a tolerant mechanism that they experience high oxidative stress. However, they can tolerate that oxidative stress, and they can still manage to get to survive longer. So I think in the roadside, it could be a similar case. Even though it's on the roadside, they have high oxidation stress, but they may have a tolerant mechanism that they tolerate the high oxidation stress. They can still manage to survive. And yeah, so in addition, it's because in the natural habitats in the roadside, there are also more other predators, insects, biological factors, ecological factors involved as well as a like community. So, yeah. Does that help your point?
Time for one more question. I have to say, oh, sorry.
Oh, there's one.
I was just going to quickly ask, do you have any idea like what kind of plants the bees are feeding on the roadside?
Yeah, so our top, we have the pollen data back, but I kind of don't want to present it if I really don't know what the data is talking about. But we know the top, on the top of the categories, actually the clover is the one of the most abundant in the pollen. Another one is the
Dill?
no, it's not dill, it's the green, what's it called? The
It's on your student's poster.
oh, Joe's poster.
No, I thought it was it Kara's poster? No,
Yes, yeah. So on Joe's poster,
Oh, that was Joe.
yeah,
Okay,
that's
sorry.
a different experiment.
Oh,
Yeah.
that's right. Okay, yeah.
Sorry.
It's been a long time.
But yeah, so some of the rose family flowers, we actually see them on the top abundance. So clover, roses, rose families, and then there's also the third one. They're green, and they're like almost like a grass type.
Sedges?
Huh?
Sedges?
Oh, fit.
No.
I don't know my Indian or Midwestern roadside.
Right, so I should study more flowers. But if you give me your email, I can like look up and send you the information.
I was just curious.
So this year, in this summer, we actually purposely collected more clover in those sites, and we wanted to test how far these clovers are from the small carpenter bee nest and what kind of pesticides we see in them. Then we can compare, maybe we'll know how far these small carpenter bees can fly. Yeah, so that's kind of another spin-off small study. But clover definitely provide a lot of pollen. If you have a yard, so I hope you can keep more clover in there as long as you can and don't.
But you're making an argument for no mow.
Well, hey,
No
you
mow
can,
May.
yeah, you can put a sign for pollinator habitat. That'll make it legit. And even their, dandelions and clover growing, I'm providing habitat for pollinators.
Hongmei, I'm going to hold my questions for later. Because of course I had something to say about cheap rating couple of receptors, so, but I'll hold them. Again, I would just like to thank Hongmei and the members of her program who've joined us here today from Central State and congratulate you again as our Osmun awardee for this year. I'd also like to say thank you to Stephen Cameron, who's the chair, and all of the members of the awards committee for their work in reviewing nominations and in selecting our awardee. So it's Paul Kamer, Matt Neidman, Amaya Gondolekar, and Taylor Nelson. So thank you very much for your service. And please join me again in congratulating Hongmei for her exceptional work on understanding honeybees and other pollinators and contributions to research and training the next generation of scientists. I invite you to, we've got, I think we've got this room until at least 5 o'clock, so you're welcome to stay, mingle, relax, socialize a little bit. We've ordered some rather swanky refreshments, so please help yourself and have a chat to Hongmei and our visitors from Central State. Thank you, and enjoy homecoming weekend.